Welcome everyone to today's webinar titled Formulating Liposomes and Importance of Vesicle Size Control. My name is Yang and I'm the New Technology Manager at Microfluidics. With me today is Professor Yvonne Perrie from the University of Strathclyde. Yvonne is a professor in drug delivery within Strathclyde Institute of Pharmacy and Biomedical Science. She obtained her PhD from the University of London, investigating the role of liposomes for drug delivery under the supervision of Professor Gregory Grigoriadis. After that, Yvonne has continued her multidisciplinary research focused on the development of drug delivery systems to facilitate the delivery of drugs and vaccines, and that's providing practical solutions for current healthcare problems. She has been collaborating with us, working on various lipid-based nanoparticle formulations, especially around liposomes. To begin today's webinar, I will give a quick overview of Microfluidics as a company, our microfluidizer technology, and processors. After that, I will turn it over to Yvonne, and she will talk about her research with liposomes, and we'll take questions and answer them in the end. Microfluidics has 35 years of experience serving the nanotechnology industry with our microfluidizer high-shear fluid processors utilizing the patented interaction chamber technology. We are headquartered in Westwood, Massachusetts, which is just outside of Boston. We have a global presence in almost 50 countries with a very large installed base. Microfluidics was acquired by IDEX Corporation and grouped with Quadro Engineering, Fitzpatrick, and Micron to form Material Processing Technology Platform. Microfluidizer processors are used for both R&D and manufacturing environments, generating critical results for many applications from pharmaceutical and biotech industries to food, nutraceutical, cosmetic, and high-performance material industries. We use our vast applications and design experience to help our customers to achieve their successes. This is about what we do best. We're very efficient generating nanoemulsions that can be filter sterilized. Our technology can also generate nano-sized liposomes, which Yvonne is going to talk about in this webinar. We also have a lot of experience working with polymer particles, including polymer nanoparticles, as well as polymer microspheres. Our microfluidizer is very efficient in cell rupture for harvesting proteins and other useful products within the cells. We have experience working with deagglomeration and particle size reduction, for example, shear exfoliating graphite to generate graphene, and also carbon nanotubes and nanocellulose. Our technology has also been used to for molecular weight reduction, for example, polysaccharide molecular weight reduction. Below here are examples of our microfluidizer processors. We offer an entire product line ranging from lab scale machines that can be scaled up linearly all the way to the production scale. How our technology works is illustrated in this schematic. The fundamental principle behind the technology is the constant pressure intensifier pump, which forces the inlet sample through the fixed geometry interaction chamber and achieve pressure as high as 30,000 psi. The system can work continuously and accommodate a wide range of materials, including high viscosities and high solid content. A heat exchanger can be used downstream the interaction chamber to regulate the temperature efficiently, so the system can work with a wide range of temperatures. We offer two types of interaction chamber designs. The one shown on the top right is a white type interaction chamber, where the inlet flow is split into two streams and forming a subsequent jet impingement. The jet impingement can generate extremely high energy dissipation rate, which in turn generates nano-sized particles for our liquid-liquid dispersion systems. That's the white type interaction chamber is more ideal for applications such as nanoemulsions, liposomes, and polymer nanoparticles. The one shown on the bottom right is a Z-type interaction chamber where there's only one microchannel and forces the flow to impinge on the solid wall downstream. This is helping for particle breakage, so the Z-type interaction chamber is more ideal for applications such as cell disruptions, deagglomerations, and particle size reductions. The other major advantage of the interaction chamber design is the linear scalability. When scaling up, we simply put a parallel array of microchannels to ensure that the velocities and shear rates and impact force and energy dissipations are identical within each individual microchannels. That's achieving a linear scalable results from the lab scale all the way to the production scale. To summarize, the microfluidizer technology is a unique technology that delivers constant processing pressure and utilizes the fixed geometry interaction chambers. The multi-slotted interaction chambers are used in the scaled-up designs. The unique technology results in the benefits of very small particle size with very narrow particle size distributions, which means you can achieve very consistent results and excellent batch-to-batch repeatabilities. The scaled-up design also guarantees the results from lab scale scaled linearly to the production scale. The microfluidizer processors are also in compliance with cGMP requirements and offer CIP/SIP capabilities. This is just a reglance through our product portfolio. We offer a wide range of microfluidizer processors, including several designs of the lab machines and a couple of pilot scale machines, including our latest offering of MF15 microfluidizer processor, and also a selection of production scale machines. These microfluidizer processors can achieve a wide range of flow rates and meet your specific requirements. So without further ado, I would hand the presentation over to Professor Perrie. Thank you very much. Here's Professor Perrie.
Thanks, Yang, for the introduction. It's great to have the opportunity to share the work we've been doing here at the University of Strathclyde. So we've been focusing a lot of our research on considering the use of liposomes for both drug and vaccine delivery. As I'm sure you all know, liposomes, because of the bilayer construct, can be used to deliver water-soluble drugs by incorporating the drug within the aqueous core, or lipid-soluble drugs by loading them into the bilayer. So when we use them for drug delivery, key attributes we consider are the lipid selection and the vesicle size. We can use liposomes to passively target drugs, improve pharmacokinetic profiles, we can design them to give slow release or drug solubilization, as I mentioned. And there's a range of products already clinically approved, such as Ambisome, Doxil, and more recently, Lipodox. So if we're going to use these systems for vaccines, then our target does change. So this time we don't want to avoid in recognition, we actually want the liposomes to target and stimulate antigen-presenting cells. We still want them to protect the antigen. We may want it to change the pharmacokinetic profile or offer delayed release of the antigen. And there are some examples of approved liposomal vaccine systems already on the market today. So liposomes are normally characterised based on the size and number of bilayers. So we can have multilamellar vesicles that are generally 100 nanometers or more. We can have large unilamellar vesicles which are about 100 nanometers but have only a single bilayer, or we can have small unilamellar vesicles which are less than 100 nanometers and are the most commonly used in the clinical products. When designing liposomes for drug delivery, it's important we consider how we control where they go in the body and the factors that might impact on their clearance. For example, if we inject them intravenously, the liposomes are recognised by the mononuclear phagocytic system, mainly the fixed macrophages in the liver and spleen, and they're rapidly cleared from the body. So avoiding uptake by the MPS is obviously very important. So there's key attributes we can consider. One of the most important ones is vesicle size, and having a small uniform particle distribution for your liposomes is very important. We tend to formulate them less than 100 nanometers, and that allows us to avoid uptake by phagocytosis. The second thing we consider is bilayer composition. So we want high transition temperature lipids and cholesterol. That gives us a nice rigid bilayer, and it helps again avoid opsonization and recognition by the MPS. The other factor we consider is the surface attributes of the liposomes, and it's well published that if we give liposomes a hydrophilic coating, such as with polyethylene glycol, we avoid opsonization. And if you look at the clearance from the blood, these are standard liposomes without pegylation. As we increase the concentration, we slowly saturate out the clearance rate, but you can see at lower concentrations we have quite rapid clearance. However, if you compare that to pegylated liposomes, you can see the dose is not having an impact on the clearance, and the clearance rate is slow because our pegylation around the surface of the liposomes is stopping opsonization, stopping recognition of the liposomes, so therefore they are not cleared by the liver. So as you can see, when designing a liposome formulation, we should take into account the bilayer design and the liposome size, as together they impact on delivery, and ultimately that will therefore impact on the activity of the system. Within our labs at Strathclyde, we're really interested in looking at the use of liposomes as vaccine adjuvants. So we've considered all the factors we've been discussing, so in terms of the vesicle size, the number of bilayers, the immunogenicity of the lipids chosen, and also the surface charge of these vesicles, and trying to look at a correlation between formulation and function as a vaccine adjuvant. So one of the formulations we've been working with for some time now was originally developed by the Staten Serum Institute from Copenhagen. So it's a combination of two lipids. We have a cationic lipid, DDA, and it's combined with a second immunostimulatory agent, trehalose dibehanate. Now, DDA on its own can form liposomes, and because of its cationic nature, it's able to carry the antigen to antigen-presenting cells. This promotes both a Th1 and Th2 type response. However, if we add in trehalose dibehanate, which cannot form liposomes on its own, but can in combination with DDA, we get stimulation of both Th1 and Th17. So we get a very robust, both humoral and cell-mediated immune responses thanks to this delivery system. When we started making these liposomes, we were using the standard lipid hydration method. So you dissolve your lipids in chloroform, you dry them down to get a lipid film, you then add buffer, warm in vortex to keep it above the transition temperature, and after about 30 minutes you form liposomal vesicles. These are cationic because they've got the DDA in them, so it means we can add negatively charged subunit antigens and they'll be absorbed onto the surface of the liposomes due to electrostatic interactions. What you can see here is a typical example of our formulation. So you could see that the vesicles are around 500 nanometers in size, and it doesn't matter if they're made with DDA on its own or we add in the TDB. They are very positive in surface charge, so they have a zeta potential about 50 millivolts. And because of this positive surface charge, we're able to stick negatively charged subunit antigens onto their surface at about 90 to 100%. So you can see in the insert we have red vesicles. That was us staining the bilayer with rhodamine, so you can see that. And then absorbed onto the surface is FITSI labelled antigen. So you see the red vesicles with the antigen absorbed to the surface. Now these vesicles, if they're made with DDA on their own, the cationic lipid, they're not particularly stable. However, the combination of DDA and TDB that makes them much more stable, and you can see the particle size stays the same over 100 days. They do have a high transition temperature, so the combination of DDA and TDB has a transition temperature of about 43 degrees C. That means when we inject them in the body, they are rigid in nature, so you've got a more solid particle type structure. So you can see we have the combination of DDA that can deliver the antigen to the antigen presenting cell and the TDB that adds further immunogenicity. So we have both delivery and adjuvant in one combination of liposome. And looking at these in small animal studies, if we compare the efficacy of the DDA-TDB liposome in combination with a TB subunit antigen, they were as effective as BCG Danish in offering protection against TB infection. So they're a very strong liposomal adjuvant. clearly had a strong liposomal vaccine system to consider. So what we wanted to look at was the interplay between the formulation of these liposomes, their biodistribution, and their efficacy as a vaccine formulation. So we started with our DDA-TDB liposomes, and then we wanted to look at the effect of charge and how if we change the charge of these cationic liposomes to neutral, what effect this would have. The effect of size, how if we played with the size, made them smaller and larger, what effect this would have on both biodistribution and efficacy. And also surface modification, if we pegylated the system, what effect would that have? So we focus mainly on biodistribution and efficacy in small animal studies. So for our biodistribution, to track our antigen, we radiolabeled our subunit antigen with iodine 125. And to track our liposomes, we radiolabeled them with tritium, allowing us to dual label and dual track simultaneously these formulations. So for our immunisation studies, mice received liposomes composed of 250 micrograms DDA and 50 micrograms of TDB. To that, we surface absorbed 2 micrograms of the fusion protein antigen 85B ESAT6, sometimes referred to as H1. This was done in collaboration with Staten Serum Institute. This was all combined together in a final volume of 50 microlitres, and mice received 3 injections at 2 weekly intervals. Over time, we collected antibody responses, and we also measured cytokine and cell proliferation. We also used these formulations and measured the biodistribution at days 1, 4 and 14, as I'd outlined on the previous slide. So the first thing we did to consider the role of charge was to formulate 2 liposome systems. So we had our standard DDA, TDB that I've been discussing, and then instead of having the cationic lipid DDA, we replaced it with a neutral lipid distereophosphatidylcholine. So we have 2 formulations, 1 cationic and 1 neutral. So if you look at this graph, the red line shows the biodistribution or the movement of our cationic liposomes from the muscle after intramuscular injection. So you can see through the red line that the cationic liposomes sit at the injection site forming a depot. If you compare that to the blue line, the DSPC-TDB liposomes, these are neutral in charge, and after injection you see them moving away from the injection site. Now our cationic liposomes can obviously electrostatically bind our anionic antigen. So if we look at how that moves, you can see again looking at the red line is the antigen stuck to the cationic liposomes. It stays at the injection site being held there. If you compare that to the blue line, which is our antigen that is associated with the neutral liposomes, there's obviously no electrostatic interaction involved, so the antigen flows away and is not held at the injection site. We also considered the impact of charge and how these liposomes move to the local lymph node, in this case a popliteal lymph node. As you can see, very low levels of the cationic liposomes reach the lymph node, with slightly higher levels of the neutral DSPC-TDB liposomes reaching the local lymph node, as shown in the red line in the left-hand graph. The right-hand graph shows how the antigen moves with it. Again, very low levels of antigen reaching the draining lymph node if it's associated with DDA-TDB, as we'd expected, because it's conjugated to the liposome and only small levels move. In contrast, we do see slightly higher levels of the antigen that was injected with the neutral liposomes reaching the lymph node, and that's because this antigen is not absorbed with the liposomes and can flow quickly to the draining lymph node. We also consider the role of liposome charge on cell-mediated immune responses. Whilst the splenocytes derived from mice immunised with antigen associated with the cationic liposomes are efficient producers of both interferon gamma, as shown in graph A, and IL-17, as shown in graph B, we can also see that the neutral formulation, the DSPC-TDB shown in grey, gave very low levels of both interferon gamma and IL-17. If we look at graph C, you can see that both the neutral and cationic liposomes were able to produce similar levels of the Th2 cytokine IL-5. If you look at graph D, again we can see that the DDA-TDB liposomes produce much higher responses and are efficient at stimulating multifunctional and central memory T cells, in contrast to the poor effector response noted by the neutral DSPC-TDB liposomes. What we can see from these results is that when we inject cationic liposomes, they form a depot at the injection site holding the antigen there. These cationic formulations also promote strong Th1, Th17 responses. If we replace a cationic lipid with a neutral lipid, these neutral liposomes flow away from the injection site, are unable to hold their antigen, and they produce low levels of Th1 or Th17 immune responses. Based on these early pharmacokinetic studies that we'd undertaken, we also thought it'd be useful to consider how important it was for us to get these cationic liposomes to the lymph node in terms of generating an immune response. So we undertook a very nice collaboration. And what you can see here is the consideration of what happens in terms of immune responses when we inject liposomes subcutaneously, the black dots, intramuscularly, intradermal, or, as shown in the triangles, directly into the lymph node. So you can see in terms of antibody responses, there's very little difference between direct injection into the lymph node, intramuscular, and intradermal. However, when we then look at our interferon gamma responses, and we take these as a key correlate of protection for the subunit antigen we're considering, you can see that the cationic liposomes give a very high response if we directly inject them into the lymph node. So this suggested to us it would be useful to consider if we could still get these cationic formulations into the lymph node after intramuscular injection to further enhance our immune responses that we were seeing in the small animal studies. So our next research question was, can we promote drainage of cationic liposomes to the lymphatics via controlling their particle size? To do this, we either produced our normal liposomes and undertook size reduction to produce smaller vesicles, or under controlled hydration we were able to produce larger vesicles. So what we're showing in this slide is, in graph A, we have the vesicle size shown in the bars with the polydispersity and values above. The grey bars are the liposomes without the addition of our subunit antigen onto the surface, and the light bars are after addition of the subunit antigen. And what we can see is we have vesicles around about 200 nanometers, with the second size, the medium, being 600 nanometers, increasing to about 1500 nanometers, and finally we have vesicles round about 4000 nanometers, with the addition of antigen making no notable difference. In terms of zeta potential, they're all cationic in nature and of a similar value, approximately 40 millivolts. Using these liposomes, we were able to consider the movement of small, medium and large DDA TDB liposomes from the injection site. So if you look at graph A, we consider the movement of the 200, 500 and 1500 nanometer liposomes at time points 6 hours, 1, 4 and 14 days. So what we can see is particle size has no impact on the movement of these liposomes from the injection site. If you look at graph B, we're considering the movement of the associated antigen from the injection site. Again, vesicle size has no effect on the clearance of antigen from the injection site. So the drainage profile after injection is similar regardless of the vesicle size. We also consider the impact of vesicle size on how these liposomes might traffic to the draining lymph node. So here we can see the data of the percentage dose of liposomes and their associated subunit antigen reaching the lymph nodes at both 6 hours and 24 hours. So at both time points we do see a trend of the larger vesicles producing a higher dose at the lymph node. However, this did not translate into the amount of subunit antigen that was able to make it to the draining lymph node. In order to examine the role of liposome size on cell-mediated immune responses, we collected the supernatants from antigen-restimulated splenocyte cultures and measured the levels using ELISA. We also took the tissue from the site of injection, digested it and analysed it for the presence of pro-inflammatory cytokine IL-1 beta. Now we did this because production of IL-1 beta has been linked to the uptake of particular adjuvants by dendritic cells. So in terms of cytokine production, we saw that vesicle size had no effect on IL-2, 5, 6, nor 10. But if you look at the graph on the left, you can see that the medium-sized liposomes were able to promote higher levels of interferon gamma compared to the other formulations. Similarly, these medium-sized liposomes, which are around about 400 to 600 nanometers in size, produced higher levels of IL-1 beta. Therefore, the vesicle size has a significant effect on the production of pro-inflammatory cytokines. So from the results so far, we were able to see that the cationic liposomes, when injected intramuscularly, formed a depot at the injection site. They also produced a strong Th1 response. Changing the size of these vesicles did not impact on this depot effect. However, we also knew that if we could get these liposomes directly into the lymph node, we were seeing higher immune responses. So how could we get our cationic vesicles moving? So we considered pegylation. Hide that cationic charge, potentially reduce aggregation, block the depot, and then we could consider what impact that has on immune responses. So how do we get our liposomes moving? We considered a combination of size and pegylation. So what you can see here is our standard MLV formulation, DDA, TDB, and we put an increase in levels of pegylated lipid into the formulation. So we've got 5, 10, 25%. This does cause a small reduction in vesicle size, dropping from about 450 to about 300 nanometers. But more importantly, you can see from the zeta potential, we're hiding that cationic charge, which was the purpose of pegylation. This didn't block too much antigen binding. We're still able to load about 80% of our antigen onto there. So it's still interacting with the cationic lipid. We repeated this with our SUV formulation, so the liposomes dropped from about 250 nanometers to about 120 nanometers. Again, we hid the cationic charge behind the pegylation. We can see that with a reduction in zeta potential, but we were still able to load about 80% or above aura antigen to the vesicle surface. So what effect did this pegylation have on the movement of our liposomes from the injection site? So what we can see here is a percentage dose of liposomes and antigen moving from the injection site with the large multilamellar vesicles. So we can see that 0 and 10% pegylated formulation have very similar profiles in terms of the liposome movement and the antigen movement. However, when we increase the peg up to 25%, we do see a marked reduction in the levels of liposomes found at the injection site even after 24 hours. So by pegylating these liposomes, we are promoting clearance from the injection site, both in terms of liposomes, as shown in the left-hand graph, and antigen, shown in the right-hand graph. Now, how does that compare to the smaller vesicles, so the SUVs? Very similar profile, so we can see that size is not having a notable effect in this case, with 0 and 10% pegylated liposomes having very similar clearance rates from the injection site, but the 25% disappearing much quicker from the injection site, both in terms of liposomes and antigen. So we know pegylation can promote movement from the injection site. How does that fare in terms of getting the liposomes to the draining lymph nodes? So here we have the levels of both liposome and antigen appearing at the local lymph node, the popliteal lymph node, after immunization. So we can see by increasing the PEG to 25%, we do see a trend of increasing levels of liposomes making it to the lymph node, though it is low in all cases. In terms of antigen movement with it, very low and no significant difference. If we look at a smaller SUV formulation, it gets much more interesting. So you can see at high levels, 25% PEG, we can certainly push higher levels of liposomes to the draining lymph node, and they do take higher levels of antigen with them. So clearly the combination of reduced size and pegylation is promoting this movement of our liposomes to the draining lymph node. So in terms of immune responses, we certainly see something interesting with the very early antibody responses. So here we have IgG1 and IgG2b levels measured in mice after 3 injections with 2 micrograms of our antigen. You can see at 12 days after injection, you get high levels of both IgG1 and 2B with the 25% PEG. We see a similar trend with the SUV and MLV. So in terms of later antibody responses, we do see some reduction by pegylation of our formulation. So again, we have our antibody responses shown here, both in IgG1 and 2B, from mice vaccinated with the various formulations. So this is 50 days after injection, and you can see in terms of IgG1, generally there's similar levels. However, if you look at the IgG2B, shown in black, the 25% pegylated SUVs and MLV have lower responses compared to the other formulations. In terms of cytokine responses, we have 4 graphs here, A showing interferon gamma, B showing IL-2, C showing IL-5, and D showing IL-6. What we can see is from interferon gamma, the DAD, ATDB, MLVs and SUVs are not actually a significant difference, but as we pegylate them 10% and 25%, we basically kill off the interferon gamma immune responses. We see a similar trend with the IL-2. With the IL-5 and IL-6, we see no notable trends with no significant difference between immune responses. So certainly our Th1 responses are reduced by pegylation. Th2 responses are not notably affected by pegylation of the liposomal system. So we'd explored a lot these cationic formulations. We tried changing the size to get them moving. We tried pegylation. Pegylation gets them to the lymph node, but it was inhibiting the immune response. So we thought, what happens if we totally reverse the charge and go from a cationic liposome formulation to an anionic liposome formulation? Can we get these liposomes moving and getting improved lymphatic targeting? So we considered 3 formulations. We had DSPC cholesterol, known to give us a nice rigid liposome formulation, and then we added either the anionic lipid dimestylphosphatidylglycerol or phosphatidylserine. So we had 2 anionic formulations and a neutral formulation. The phosphatidylserine PS formulation has a high transition temperature, about 6 to 8, whereas the DMPG has a transition temperature at 23. So the PS formulation will be rigid in the body, whereas the PG formulation will be fluid light. So we were effectively looking at both charge and fluidity. We made the liposomes again by the standard lipid hydration technique, but obviously this would produce large liposomes, so we needed to consider how to reduce the size. These vesicles were also radio-labelled. So to produce our small unilamellar vesicles, we were looking at using the high shear processing system that Yang has already outlined. So we were using the Microfluidics M110P. And the options this is, we could vary the operating pressure, the number of passes and the temperature so that we could optimise the size reduction and produce small unilamellar vesicles. So we started looking at liposomes with low transition temperature. So these are the PC-based formulations. And what you can see in the left-hand graph is we rapidly have size reduction. After only 3 passes, our vesicles are down to about 60 nanometers. The operating pressure can be varied between 20 and 30 kpsi without any impact on the efficacy of size reduction. Our PDI, or dispersity of these vesicles, is low, so we have a nice compact size range, and that's shown by the right-hand graph as well. So then we also tested with high transition temperature lipids, so this is DSPC, cholesterol-based formulations. So we see a very similar trend. So again, we've got very effective size reduction rapidly. So after only 3 cycles, our liposomes are coming down into the SUV range, around about 60 to 70 nanometers. We can use at a range of pressures, and the PDI is again small, with a single population of small size, small unilamellar vesicles. So now comparing the effect of size with anionic liposomes, we have 2 formulations here. One is DSPC cholesterol PS, which is in the blue line. So these are the small unilamellar vesicles, and the red line is our large multilamellar vesicles before size reduction. So if you look at the dose at this injection site, you see our larger vesicles stay at the injection site and take some time to drain away, whereas our small vesicles rapidly drain away from the injection site. And where do they go? Well, if you look at the right-hand graph, you can see that the small vesicles are moving away from the injection site and appearing at our draining lymph node, approximately 24 hours, and we get around 30% of the dose appearing after that time. So that was great. We are able to move these liposomes to the draining lymph node. So we knew our small anionic vesicles were nicely moving to the draining lymph node, so we thought it was useful to do a head-to-head study between small neutral liposomes, as in DSPC cholesterol, compared to those containing phosphatidylserine, as shown in the second line, again very negatively charged, minus 66 millivolts. We have a similarly negatively charged formulation containing DMPG. And then finally, we have a control of a cationic formulation. So we used, in this case, diolutrimethyl ammonium propane just to give us an alternative, but these are very cationic in nature. So they're all around about 90 to 100 nanometers in size, with PDIs below 0.2 generally. In terms of drainage from the injection site, we see that our cationic formulation, shown in purple, sits in the injection site as we've seen it with previous formulations. So the cationics are aggregating and sitting there. Then if we compare our blue, green and red lines, so our blue is our neutral formulation, no aggregation, it's clearing, and it's clearing at similar rates to our anionic formulations. But what you do see if you compare between our two anionic formulations, the red line, which contains phosphatidylserine in the liposomes, is cleared at a faster rate than the alternative anionic liposomes, despite them both being of a similar size and similar anionic charge. So the choice of lipid is having an impact on how these liposomes flow from the injection site. So if we now look at how much these liposomes are able to move to the lymph node, we can see that our small SUVs containing phosphatidylserine are able to track to the highest level to the lymph node, whereas our neutral formulation and our cationic give lower levels, as does our alternative anionic liposome formulation. So it's not just charge and size, it's also the type of lipid within the formulation that has an important role in the draining to the local lymph node. We're seeing differences in these clearance rates from the injection site and appearance at the lymph node with these two anionic formulations. We wanted to know if there was a cell-mediated component to this. So were the liposomes being taken up by preferentially and being moved to the lymphatics. So to investigate macrophage preferences, what we did was we labelled our liposomes with DIL-C, so a fluorescent marker. We then incubated these with macrophages or DCs, and we measured the uptake of the liposomes via fluorescence. So using these uptake studies, we do actually see macrophages have a preference for small phosphatidylserine containing liposomes. So if you look at the graph on the left, you can see there is high levels of liposome positive cells when they are treated with small unilamellar vesicles containing phosphatidylserine. And the mean fluorescent intensity of macrophages is again higher when the macrophages have been treated with small unilamellar vesicles containing phosphatidylserine. In summary, if we want to promote lymphatic targeting, we should use SUV liposomes, which we can very nicely produce using the Microfluidics M110 system, as we've shown in this talk today. We should incorporate an anionic lipid, particularly phosphatidylserine, which promotes drainage to the lymphatics, and we can exploit this for a range of both drug delivery to the lymphatics, or in our case, we've been looking at it as a potential vaccine adjuvant delivery system. Finally, I would just like to introduce you to the team here at Strathclyde who undertook the work, and also to gratefully acknowledge the strong collaboration we've had for many years with the Staten Serum Institute, who we've worked with in all these studies, and also with our colleague Professor Andrew Devitt at Aston University. Finally, obviously, we'd like to thank our sponsors, and I would like to thank you for your time listening today. Thank you.
Thank you again, Yvonne, for the excellent presentation. We're now open for questions. We actually have already received quite a few questions already. The first question is, can you comment on the factors that affect the stability on storage and shelf life of liposomes?
Yeah, that's a good question, Yang, particularly with the smaller vesicles that we've been making here. So generally your choice of lipid has an impact on their ability for stability. So we have to think chemical stability? Do they oxidize? And quite a lot of them do. But if we go for the higher transition temperature lipids like the distereophosphatidylcholines and combine it with cholesterol at a 50-50 ratio, molar ratio is generally good. Those tend to be our most stable and can hold the drug well inside them. But for long-term storage, most of the time we go for a freeze-dried product because it's easier to store longer period. And you see most of the products on the market are freeze-dried products, so we freeze-dry them with a cryoprotectant or a lipoprotectant in there, and that helps reconstitute. So they're normally stable in the fridge for, depending on what you made them from, easily up to about a month. But after that, it's probably best to consider, if it's for shelf life, freeze-drying the product.
Thank you. The next question is, what is the smallest size of liposomes that can be produced?
Yeah, that's a good question too. So theoretically, if you do the calculation, the absolute minimum size we could get would be a 25 nanometer vesicle. I don't think that's actually achievable because that's just based on adding up the actual size per molecule. So with the microfluidic systems, we've been getting around about 35, 40 nanometers down to that size range easily. If you put a charged lipid, sometimes that causes them to go slightly bigger because the electrostatic repulsion between the head group means you can't compact down the bilayer as small as you might like. So, but yeah, 35, 40 is now an achievable size range, which you wouldn't be able to achieve with the old-fashioned sonication. We could never get that low before. We were maximum 80 nanometers, minimum 80 nanometers we could achieve.
Thank you. The next question is, I'm very much interested in knowing all the parameters controlling vesicle size. What is the effect of drug molecule size on encapsulation, and what effect does encapsulating drug in lipid bilayer have on flexibility of liposomes?
Oh gosh, yes, good question. So in terms of the factors that control the size, the vesicle size that we can get, so that is obviously dependent on our choice of lipid, our concentration of lipid to some extent, though in the concentration ranges we've been working with at the moment with the system, we haven't seen an impact on that. So it tends to be the higher transition temperature lipids. You may need to put them through an additional pass, but generally most of the time we're getting them all down to the same size. But technically the questions you would ask are, what is the transition temperature of my lipid? What is the concentration of my lipid? Because if it's a very high concentration, it will need more energy for size reduction. And do I have cholesterol in it, which can, again, as I say, have an impact on it. In terms of the effect of drug molecule size encapsulation, if it's a water-soluble drug, it won't really have any impact as far as we've seen in our data. So we've used small molecules right up to large proteins, and they don't have an impact on the vesicle size. If you put the drug within the bilayer, that can influence the liposome size because it impacts on packing of the bilayer. So sometimes we were seeing them increasing in size slightly if we were putting it in the bilayer. But again, we could get particle sizes about 50 nanometers using the microfluidics. And in terms of the last part was encapsulating the drug in the bilayer has on flexibility of liposomes. We haven't really looked at that too much. It would depend. So we can, certain drugs, if they have a cholesterol type structure, would impact on the fluidity of the bilayer in the same way as cholesterol does. So anything that can fill in those gaps between the phosphatidylcholine tails would impact on fluidity. And I guess you can measure that through extrusion processes to see how easily they can be extruded after being prepared, because that's quite an easy way to measure fluidity of a bilayer.
Great. I just want to add on a couple of points regarding using microfluidizer processor to produce liposomes. As we already pointed out in the presentation, there's several factors we can control with the processor, which are pressure and number of passes and temperature. And temperature is very critical, so you want to process above the phase transition temperature, and also pre-mixing is a very important step. We have here at Microfluidics, we have a lot of experience. We have an excellent application team, and we have a lab here, so we can work with you to help optimize your formulation and process. The next question is, what is the best way to characterize vesicle size?
Okay, so we use dynamic light scattering, and we always use that in combination with a second method. So at the moment, the regulators are normally, they'll accept dynamic light scattering, but there is a debate whether we now need a second method. So we tend to back up with cryo-TEM for these smaller vesicles. So that's the one that gives the least change in size we find, through manipulation, because we're not drying it out as such because we're freezing it. So we would always say some sort of dynamic light scattering, whatever system you want to use in that respect, but back it up with a second method so that isn't just a DLS again. So microscopy is very useful, and to get down to the size range, it's normally cryo-TEM that gives you the best, or freeze fracture is another one.
Great. The next question is, what are the most important factors to consider when designing a drug release medium and mechanism?
Okay, so to consider how well your drug's releasing out, you consider the attributes of the drug. So if it's a water-soluble drug, then that's easy. You just go with a biologically relevant media. So if you want to consider how it would release after IV injection, then do release into plasma. If you've got a low solubility drug, you have to make sure that the drug can actually release into something, because you have to maintain sink conditions so you have that diffusion gradient. So in that respect, when it's low solubility, you might have to play around with the dissolution media. Some people use cyclodextrins or other solvents to help the drug dissolve once it comes out of the liposome. So that's the kind of two things. There was a nice paper recently. Frank Zoka did some work looking at release of doxorubicin and trying to have an easy marker for it, and that's obviously water soluble. So there's been some nice papers recently looking at that and various dissolution setups that we can use now.
Thank you. The next question is, what characteristics should the phospholipid carrier have? Is it possible to use less pure phospholipids?
Okay, yeah, I mean, certainly that can be done. It really depends what your target for your product is in terms of the market. So obviously if it's going IV, the regulatory requirements are quite strict. So obviously you need to be able to validate your product in terms of quality control and quality assurance. The more pure your product is that goes into the liposome, the easier it is to quantify. But certainly, I mean, you can make liposomes with 60% pure phosphatidylcholine. The cheaper stuff is very easy, and it will still work fine. I guess it just depends what you want to use the liposomes for, but they do form with less pure lipids, no problem.
All right, I have one more question. It's regarding formation of cationic liposomes. Do you know if purified phosphatidylcholine such as Leciva S90 will form cationic liposomes?
I'm not familiar with that lipid as it is, but generally yes. Most lipids, if you put in cationic lipids, they can form. The key is to consider your composition. So overall, obviously, the difference between a micelle and a liposome is based on the geometry of the surfactants going in. And our phosphatidylcholine, for example, because they have two tails, biases to the bilayer structure. Quite a lot of the cationics, because they have a very large head group, because of electrostatics, means their overall shape is quite wedge-like, and will try to push the liposome back to a micelle type structure. So as long as you compensate for the large head group of the cationic lipid with other lipids, then yes, you can do that, no problem. So it's not a problem having a combination, as long as you make sure you don't put too much of the cationic lipid in, because then it can actually revert over to a micelle rather than a liposome.
Thank you. Can you comment on this question? Does the charge of the liposomes produced remain intact after microfluidics processing?
Yes, so yes it does. So we have put cationics and more anionic. We've been working with that recently as we outlined in the paper, but definitely yes, it stays intact, no problem. so there is no damage to the lipid or anything like that. So yes, we get out the system a very negatively charged vesicles around about minus 60, minus 70 millivolts, and they are very small in size. So yes, it's not a problem at all. The charge remains intact after processing.
Thank you. I see no more questions coming in. I think I will just conclude today's webinar. Thank you, Yvonne, again for the excellent presentation, and thanks everyone for joining us today.
Thanks for that. I'm glad people were able to phone in for it.
Thank you.